All right, welcome. So these are our Mendel and Genetics notes. These are quite long notes, so if you need to, go ahead and write them down, and then you can get them into the actual Cornell notes later, okay? It is very, very, very important that you watch this entire video. All right, so Mendel and Genetics notes. Your Cornell template is here. It's under Unit 5. All right, so let's go ahead and get started. All right, so Mendel and Genetics. Mendel is who we call the father of genetics. And so you'll learn a little bit about his experiments here, and then we'll go into how to use what he discovered. Okay, so our essential question here is, how can Punnett squares help to determine the phenotype of offspring? And of course, there's a ton of vocabulary in there. Remember, phenotype P is for physical appearance, and the Punnett square you'll get used to as we practice with them. Okay, so what is genetics? Well, genetics is a scientific study of heredity, and we use it in humans when we want to track maybe an undesirable trait, maybe something that is like cystic fibrosis or Down syndrome, which I studied up in Stanford. They believe that has a genetic point to it, but we can track and see if there's some kind of things in our DNA that cause us to have those diseases, okay? And then what is a trait? Well, a trait is a specific characteristic that varies from one individual to another. So example like brown hair, blue eyes, tall, curly hair, all those things are different traits, and they are controlled by the proteins that are made by our DNA. Okay, and then what is an allele? Allele is very specific. So alleles are the different possibilities for a given trait. So remember our little sister chromatids here, and now we're going to use letters to represent alleles. So each letter is an allele, and basically it's one copy of a gene. So every trait has at least two alleles, two copies of the gene, one from the mother and one from the father. So example, eye color. So examples of alleles, like a big A would give us brown eyes. So since this has this chromatids here have a uppercase A and uppercase A, this would be brown eyes because there is no blue eyes, okay? And then this one here, a little a, these would code for the proteins to make blue eyes, okay? And we'll learn what happens when we have an uppercase A and a lowercase a together, okay? And then what are genes? Genes are the sequence of DNA that codes for a protein and thus determines a trait. So basically, remember when we talked about protein synthesis, those genes that we said when we had the codons and the anticodons, those three bases, is what codes for certain proteins that makes your hair color the color it is and your eye color the color it is, okay? So Gregor Mendel actually started this whole discovery. He is what we call the father of genetics, and he did the first important studies of heredity, how things are studied. And he identified specific traits in the garden pea plant and studied them from one generation to another. So he didn't study humans, he actually studied pea plants. So let's look. So his conclusions were that things of a law of segregation. So he realized that there must have been two copies and that there's a copy from the mother and a copy from the father, and they separate by themselves. So remember in meiosis, when your gametes are made, you have your 46 chromosomes, but then in the gametes, we only have 23 because we only have one copy. And that's how we call this law of segregation. So two alleles for each trait separate when gametes form. Parents pass only one allele for each trait to each offspring. So just like we looked at the two uppercase A's, one uppercase A would go in one gamete, the other uppercase A would go in the other gamete. And then he also discovered the law of independent assortment. That means genes for different traits are inherited independently of each other. That means all genes are random. They go in their own order. Now, you may learn as you get up higher in biology, this doesn't always happen. We do have some that we'll talk about like X-linked traits, but most of the time it is independent assortment, okay? And then we have two other things that we talk about. Dominant, which basically it masks the other trait, the trait that shows if it's present, and we always represent it with a capital R or a capital letter, okay? And that just means no matter what letter is with it on the other chromatin, then it is the one that is seen. And then there's recessive. An organism with a recessive allele for a particular trait will only exhibit that trait when the dominant allele is not present. So if we don't have an uppercase, then we'll see the trait with the recessive. And we'll only show both alleles are present, which means both of the alleles are recessive. And it is represented by a lowercase letter. Okay? So here's some examples. So we have dominant and recessive practice. So let's say uppercase T says straight hair and lowercase t says curly hair. And any offspring with two uppercase T, of course, would have straight hair because we have both dominant trait alleles there. But what if an offspring has an uppercase and a lowercase? Well, since our uppercase is present, it is dominant, so it will take over and it will mask the lowercase t. So that will also have straight hair. The only way you get curly hair is if you have two lowercase t's, and that's what we call recessive. And this is what we call a homozygous dominant. This is a heterozygous, and this is a homozygous recessive. And we'll talk more about that as we practice this, okay? How about genotype versus phenotype? So the genotype is a genetic makeup of an organism. So the gene or allele. So it is our letters, okay? That is a genotype. That is what our genes tell us. But a phenotype is the physical, see the P, physical characteristics of an organism. It's the way it looks. So curly hair, straight hair, blue eyes. So as you can see here, we have a purple flower here. That is the phenotype. That is what I can see. But its genotype is two uppercase A's or uppercase A, lowercase a. And I can tell you that because this purple shows up in both of these, then it is purple is dominant. Well, white is recessive because it only shows up when there's two lowercase a's, okay? And then you have just what I was talking about, homozygous versus heterozygous. Okay, homo, H-O-M-O, that is a Greek word. It means the same. And hetero, H-E-T-E-R-O, that means different. So those are just root words from Greek words that tell you the same or different. So homozygous term is used to refer to an organism that has two identical alleles. So either two uppercase R's or two lowercase letters. And a heterozygous is used for having different ones. So an uppercase and a lowercase together. Okay. Now let's look at our Punnett squares. So this is what Mendel kind of designed. Coming out to figure out what kind of genotypes and phenotypes would come out of a cross. And we actually do use this. I actually use this when I did research in genetics. We use it all the time. So you can see it's a diagram showing the gene combinations that might result from a genetic cross and used to calculate the probability of inheriting a particular trait. Remember, probability is a chance that a given event will occur. So if I had genes and I had two known genotypes that were heterozygous, so I had uppercase A, lowercase a times uppercase A, lowercase a. Now, we talked about the law of segregation and law of independent assortment. No other genes are coming with it. We are just only going to determine this gene because it doesn't depend on what else comes with it. And then independent assortment means these will separate on their own. So that's why you see these squares. So we put an uppercase A here. This can be the mother or father with a lowercase, and then the other parent goes here, uppercase and lowercase. So they can independently sort, and they will segregate. Then you will just fill in the boxes. So you'll bring this A here with this other parent here, and then this one also goes here to bring this one down here. So notice they can combine this way. Or this one can come in with this one, and this one can come in with this one. So this gives me all the different types of genotypes that I could get, okay? All right, so you can see you have your parent, and these all in the boxes would be your offspring, okay? All right, so as I just explained, you will have the beginning come down, this one will come in, this one will go to here and here, this one will go to here all the way across, this one will go all the way across as this one goes all the way down, okay? All right, so that's how you complete a one gene Punnett square. So let's look at a couple. So one of the things that Mendel looked at is the color of pea plants, and this is how he kind of figured it out. So each parent could give either a Y, an uppercase Y, or a lowercase y, based on the two genotypes. So he crossed yellow plants, because that is dominant, together, and he found something, okay? He found that sometimes he got white. Why would that happen? Well, that's because of our Punnett square. So if we put our one parent here and one parent here, so each one can give either an uppercase Y or a lowercase y, and then we fill in our boxes. This uppercase goes here and here, this lowercase goes here and here, this one goes here and here, and this one goes here and here. And notice what we get is 3 different genotypes. So we could have homozygous dominant, we could have heterozygous, or we could have homozygous recessive. And the way we show it is that we could have one here, okay? We could have 2 of these and 1 of these. Just like here, this would be split up into 25%. So I would have 25% homozygous dominant. I would have 50% heterozygous, because 25 plus 25, and then 25% homozygous recessive. And I would see 3 yellows, 1, 2, 3, to 1 white every time I would cross them. Okay, so let's try it now. Okay, we're going to use the genotype for tall. So we're going to have a homozygous dominant and a homozygous recessive for tall and short. And he did these for the pea plants as well. So first thing you do is set up the Punnett square. So we have our T and T for one parent, uppercase T's here, dominant, and then the recessive here. And then the next thing you're going to do is you're going to start cross them, is complete the Punnett square. So you fill in the first one, this T here joined with this T, and then this T joins with this T, and then we fill in next box. This T comes down, this one comes over, this one comes down, comes over. Now notice, because we have maybe 2 different genotypes in the parents, and one is tall, this one would be tall, and this is short, we're going to have the same genotype for all of the offspring. So they're all going to have the same genotype, and they're all going to have the same phenotype. And remember, each box is worth 25%. All right, let's try another one. What if we had a heterozygous times a homozygous recessive? So we'll put in our letters, and then we'll start crossing them, okay? So that will get us a heterozygous. This one, though, will give us a homozygous, heterozygous, and homozygous. Now, see what our genotypes are. So we have heterozygous. We're going to have 2 of them here. So 50% heterozygous and 50% homozygous. And the phenotypes is we're going to have 50% tall and 50% short. Okay. What happens when we have multiple alleles? So 3 or more alleles of the same gene. And we're not going to go all the way up to 3. We'll just go to 2 here in this class. And even though 3 or more alleles exist for a particular trait, an individual can only have 2 alleles, 1 from each mother and 1 from each father. So you can see this is an example of chinchilla. So you can have different mixing of different colors. And I'll show you how to do those, okay? Okay, so coat color in rabbits is determined by a single gene that has at least 4 different alleles. Crazy, we won't go that far. Different combinations of alleles result in the 4 different colors that you see, okay, here. So let's look. So another one that's a little easier to see is blood type. So we have all kinds of different blood types. We have A, we have B, we have O, we have positive, we have negative. It all mixes together, okay? And so which results in 4 different possible blood types. And so what happens, and we're going to practice this in class, but what happens is you might have an A and you have a B, or you might have what they call an I, which is O. And then you can mix them. You can have an A, so this would be all A. You could have an A and a B. You could have an A and here an I, a negative. You could have a B here, just B, B negative, and then you have A negative, okay, and you have B negative, and then you have O here, I, okay? And so all of this kind of mixes together. Now, notice the I is lowercase, and that's because O is very rare, but O is also recessive. So if any of the other ones are present, then you just end up with the first letter. Such as with me, both my parents are A, and so I ended up with 2 A's, okay? I might have one of these in here, an A and an O together, possibly somewhere, I'm not sure, but I don't know exactly which one I am. But if you're an A and a B, that means A came from your either mother or father, and the B came from your other parent, okay? And to be O, you have to get some kind of O from there. And so one of the things we're going to learn is pedigrees. And you can track my pedigrees if you want. You can see how mine works because I am A and my husband is O. And so I know that his genotype is here. And I know that possibly mine is most likely this one over this one. And the reason is because all my kids are O's. So the only way they could have done it is if I gave them a gene for that O somewhere in there, and he gave them one as well. If I was this genotype, all my kids would be A, but they're all O. It's kind of strange. Okay? All right. There are always multiple alleles. So genetic inheritance is often represented with straightforward examples involving 2 alleles with clear-cut dominance, and it's not always the same. This makes inheritance patterns easy to see, but very few traits actually only have 2 alleles with clear-cut dominance. As we learn more about genetics, we have found that there are often hundreds of alleles for a particular gene, and we probably already know this, but as we look around each other, we see infinite variation. So even such as here you see skin color. Maybe even in my own family, one of the things, I'm very light skinned as my other kids. 2 of them are very light skinned like me. Other ones are more like my sister, like a little bit darker. So we're of the same family, but we all have different shades of skin. And so we call those a polygenic. So poly, P-O-L-Y, means many. And it is a trait controlled by 2 or more genes. And polygenics often show a wide range of phenotypes. And you might look around your family and see that. And the examples of wide range of skin color in humans comes about partly because more than 4 different genes probably control this trait. We have no idea how many genes actually control it. We just know it's definitely more than 1. Okay? So remember we talked about just a minute ago about but sometimes we don't have complete dominance. And so I'm going to talk about a couple different things, and we'll practice these in class. But the first one that doesn't say complete dominance, where it's just straightforward, is what we call incomplete dominance. And it's a situation in which one allele is not completely dominant over another. So one example is that Mendel found is that when he crossed some red flowers, so some white flowers, he didn't get red and white. He got pink. And so that just shows that the red and white mix, which means you kind of see them together. They kind of mix in together. So maybe you could have something like a black bear and a white bear, and they have offspring, and they end up gray. And it's just a mixture of the 2 phenotypes that you started with. So incomplete dominance means that it's like a mixture. Codominance is completely different. So codominance is a situation in which both alleles of a gene contribute to the phenotype of the organism. So one of the things that I saw and many times is what we call a rowan cow. And so it's from mating 2 cows together, 2 different breeds. And so you can see them here. And one of them's a white breed. I won't tell you their names. And the other one is a brown breed. And when you cross them together, we got something that looks like this. A white and brown spotted cow. And what that means is we call them rowan, and what that means is they have a codominance. So when we get that heterozygote with the, let's say, big B, little b, then what happens is you see both phenotypes. Not a mixture of the phenotypes, but you see both of them. So you could have things like, just like I said, the black bear and a white bear, but you could also have, sorry, that was the bell, but you could also have situations where it's a black and white spotted bear, and that would be more of codominance, okay? All right, so that's the end of our notes. So highlight any important notes, make connections to any of your notes in the left-hand column, write any questions you may have in the left-hand column, and use your highlights, connections, questions for your summary. And at the end, make sure you're writing a 3 to 5 sentence summary. And if you have any questions, please let me know.